Thank you all for coming. Good morning. My name is Ziervogel, and I am Lydia Pinard's research advisor. And I welcome you to Lydia's thesis defense. Today, Lydia is pursuing a master's in oceanography here at UNH. And before I let her talk about her research program and her thesis, I'll give a little bit of a background here for you all. I need to turn this around. Okay, so Lydia came to us from the University of New England, where she got her bachelor's in marine sciences in 2022. And right after that, you came over and you started your UNH experience. Pretty extraordinary, I should say, because before classes started, we had her come with us on a research expedition in the Gulf of Maine and Nova Scotia or Scotian Shelf, led by UNH by Dr. Joe Salisbury, who's here. He's Joe's one part of Lydia's committee, as well as Dr. Elizabeth Harvey over there. Lydia was a TA, a teacher's teaching assistant, throughout the entire 2 years of her life as a graduate student. But she also experienced additional research opportunities, like last summer, where she was at sea for almost 2 months.
Yep.
At a very exciting, extraordinary experience, sailing from Australia to South Africa on a US ghost ship expedition. And that stands for, and I had to write this down because I will never remember this, is the US Global Ocean Ship-Based Hydrographic Investigations Program. And that was a national call for participation, and Lydia applied, she won, and she was able to go out. And to me, really, the graduate experience here at UNH in oceanography includes being out at sea and experience the life of an oceanographer in the field. And you got a lot of it, actually, in these 2 years. So now, please tell us about your research. The presentation will be about half an hour, 40 minutes. And then after that, we all have the chance to ask questions. And then the committee will convene with you, and we'll take it from there. Thank you very much.
Sounds great. Thank you so much for that wonderful introduction, Kai. And thank you all for attending my defense this morning, both virtually and in person. Once again, my name is Lydia Pinard, and I'm a master's student here working in the Ocean Process Analysis Laboratory, or OPAL, here at UNH. Just want to take a second. Can everybody hear and see me in person over Zoom? Not hearing anyone saying no over Zoom, so that's great. Before we get started, I would like to reintroduce my committee, Dr. Ziervogel, my thesis advisor and director of OPAL, Dr. Elizabeth Harvey, Associate Professor of Biological Sciences here at UNH, and Dr. Joseph Salisbury, Research Professor, Earth Science Affiliate and member of OPAL. This morning, I will be sharing my graduate research on the impacts of warming waters on phytoplankton respiration. I also understand that there are theses right after this. So if you guys need to head out early, I completely understand. And we will continue on. You have a clicker. It is working. So the schedule for this morning is as follows. I will start by introducing you all to the net community respiration of marine phytoplankton, as well as the effects of climate change and sea surface warming on net community respiration. Then we will discuss my methods, which are broken into fieldwork and laboratory methods using a specific species of diatom and copolithophore. I will then contextualize these findings with my hypotheses, and finally there will be time for questions. Before we jump into my methods, I'd like to introduce you all to a few key concepts. The first being cellular respiration. Cellular respiration is the process that converts biomass and oxygen, as you can see in the equation here, into energy within cells. Cellular respiration plays a very important role in both biological and microbial carbon pumps in the ocean. As you can see in this schematic here, respiration drives oxygen consumption and conversion of organic carbon pools in the form of refractile DOC into carbon dioxide and its emission into the atmosphere. Because of this, respiration is an indicator of carbon budgets in marine systems. In the pelagic or open ocean environments, 34% of respiration is performed by phytoplankton. Because of this high percentage, we can use respiration rates to study the metabolic state, including growth rate, abundance, and mortality of phytoplankton communities. Respiration is sensitive to temperature change, and Garcia Martin and Pascalo Galazza predicted that as seawater temperatures increase, so will phytoplankton respiration rates. As respiration rates increase, CO2 production and dissolved inorganic carbon, or DIC, production increases, and pH will decrease. This will further exacerbate anthropogenic or human-caused climate change. Thus, cellular respiration rates can be used to predict biogeochemical responses to climate change. Phytoplankton are responsible for half of the global primary productivity, which is the creation of new organic matter from inorganic substrates that serve as the base of the food chain for most marine consumers. They do this annually. As mentioned briefly in my previous slide, phytoplankton have crucial roles in the biogeochemical cycles within the ocean. Across the functional groups, marine phytoplankton contribute to nitrogen fixation, carbon sequestration, and the cycling of phosphorus and silica. The four major types of phytoplankton are cyanobacteria, dinoflagellates, diatoms, and coccolithophores. Cyanobacteria are probiotic phytoplankton responsible for the evolution of oxygenic photosynthesis. In modern times, these phytoplankton now contribute significantly to global natural nitrogen fixation. Dinoflagellates are large and diverse phytoplankton that have complex traits including motility, bioluminescence, and toxin production. Diatoms are large silica-rich phytoplankton that vary in shapes and sizes. In this image here, these are all different diatoms. They perform 20 to 25% of global primary productivity, and they're involved in deep ocean carbon export, as well as the burial of silica at the deep ocean. Coccolithophores, which you can see a few different coccolithophores here, they have these unique calcium carbonate plates on them. That's these little shells that they have on top of them. And they are also responsible for approximately 10% of the carbon export to the deep ocean. For my particular research in my lab setting, I chose to focus exclusively on diatoms and coccolithophores. The global ocean is a carbon sink, and according to the Intergovernmental Panel on Climate Change, the uptake rate of atmospheric carbon dioxide in the ocean has increased in recent decades. Anthropogenic or human-caused climate change has taken many forms in the ocean, including changing chemistry, anoxic and hypoxic conditions, ecological change, sea level rise, changes in currents, ocean acidification, and warming waters. For this study, we're focusing on the response to increased water temperatures. When organisms are not used to warmer temperatures, they're forced to either move to another environment, evolve very quickly, or die out. I want to share one of my favorite pieces of art with you all that shows the warming in the Gulf of Maine. This is by a local artist in Portland named Jill Pelto, and it actually shows real data of the Gulf of Maine sea surface temperatures increasing. So all of these here are actually real data points in the Gulf of Maine. And it shows the movement of the Atlantic cod and the American lobster northward to reach cooler waters as the waters are warming. The Intergovernmental Panel on Climate Change has predicted that the Atlantic Ocean will have a temperature increase of 1 to 4 degrees Celsius by 2100. You can see in this figure here, even in the blue range, which is the most moderate scenario where we stop producing greenhouse gases by 2040, there's at least going to be an increase in the Atlantic of 1 degree Celsius. On a local scale, the Gulf of Maine is warming faster than 99% of the global ocean. By observing the changes in the Gulf of Maine and what they're currently facing as a result of climate change, it may be possible to predict future changes in the global ocean. I have two major hypotheses for my work. I'll give you a second to read them before I go into them. The first is that net community respiration in the Gulf of Maine in the summer is dominated by the size class that represents phytoplankton. Additionally, that the net community respiration in the Gulf of Maine in the summer differs on both a spatial scale and with depth. The second hypothesis for my lab experiments is that increasing water temperatures will lead to increased rates of phytoplankton growth and respiration. This was tested specifically using cultures of Cattosaurus tenuissimus and Emiliana huxleyi. On to my methods. First, we will be discussing my laboratory experiments. The two species of phytoplankton I chose for my lab experiments are Cattosaurus tenuissimus and Emiliana huxleyi, or E. huxleyi. I have included images of them both very close up under a microscope and also in our microscope downstairs at 10x on a hemocytometer. So initially we counted these by hand, and so all we would have to do to count our cells to make sure they were growing properly was find these little guys, our little squares for C. tenuissimus and our little circles for Emiliana huxleyi. We ultimately ended up using more high-tech ways to count these little guys. C. tenuissimus is a chain-building diatom with a global distribution, and it is also adaptable to warm waters. Emiliana huxleyi is a frequently studied coccolithophore with global distribution and a wide temperature range, able to survive from 2 degrees Celsius waters to 27 degrees Celsius waters. Both species are prevalent within the North Atlantic. Cultures were provided by the UNH Phyto Lab courtesy of Dr. Elizabeth Harvey. 3 temperature treatments were tested. First was the 19 degrees Celsius treatment in a temperature controlled room on the first floor of this building. There was also a 22 degrees Celsius temperature treatment in the Ziervogel lab and a 30 degrees Celsius treatment in an incubator in the UNH Phyto Lab. All treatments were under lights that emitted 80 to 100 micromole photon per meter squared per second. Unfortunately, due to some constraints in accessing rooms to grow our cultures in, the 19 degrees Celsius and the 22 degrees Celsius treatments had constant light 24/7, while the 30 degrees Celsius treatment had a 12 on, 12 off light cycle. All phytoplankton were cultivated at 19 degrees Celsius in F2 medium with nutrients and at 35 parts per thousand for the salinity. Cultures were then moved to their new temperature conditions and had 10 days to acclimate into their new temperature environment, which is a full growth cycle for both species. This was done prior to our experimentation. It should also be noted that a flask of just our F2 medium was kept in all 3 temperature conditions and underwent our experimentation to serve as a control. Prior to undergoing our respiration experiments, we made sure that all phytoplankton were in exponential growth, as this is when the cultures have the highest cell abundance. This was determined by performing daily cell counts using the UNH Phyto Lab FlowCam. This instrument runs water samples and is able to actually count the cells for me, so I don't have to do it by hand, which was much appreciated. The growth rate was then calculated using this equation here, where K prime is equal to the growth rate in cells per milliliter per day. T2 and T1 are time points in days, where T1 is the first day that cell counts were conducted, and T2 is when our experiment was actually conducted. This varied per culture. It took the different samples longer or shorter periods of time to get to exponential growth. Generally, it was between 2 and 4 days. Additionally, N1 and N2 are the biomasses in cells per milliliter, where the cell abundance at time points 1 and 2. Exponential growth was determined by whether the cultures had either doubled overnight or increased by an order of magnitude overnight. While the established method of measuring respiration is the 1888 Winkler method, this requires long incubations and processing time, as well as multiple substrates being added to water samples. So we decided to focus on a new method, the newly established in vivo electron transport system, which is done on living phytoplankton cells. This electron transport system activity method requires shorter incubation times and processing time, one substrate, and also has high sensitivity. Additionally, something really unique that I found really interesting about this method is we actually are able to determine what sizes of plankton are undergoing the respiration. This is not possible with the Winkler method. As previously mentioned, the in vivo ETS method requires one substrate. This is tetrazolium salt 2-para-idophenyl-3-nitrophenyl. A little bit of a mouthful, so we tend to call it INT. This INT is a redox dye that replaces oxygen as the terminal electron acceptor in the electron transport chain. The electron transport chain is one of the last portions of cellular respiration. And as you can see here, the oxygen serves as the last electron acceptor to actually produce ATP. And what happens here is the INT takes the place there and it crystallizes to become formazan. Now, the interesting thing about this is that once the substrate is actually added and is converted to formazan, we can actually measure the absorbance of the sample to detect the presence of the crystallized form, or formazan. And therefore, we can figure out how much oxygen has been consumed in respiration. To measure respiration, samples were taken from all cultures, including our control. They were put in total darkness for 20 minutes to ensure that only cellular respiration was occurring, not photosynthesis. And after that time period, the INT substrate was added. After the addition of INT, the samples were then incubated in the dark for 2 hours to undergo respiration. Samples were then neutralized to stop respiration and then filtered through a pump equipped with a 2-micrometer filter. These filters were then collected and processed, and the absorbances of each sample were recorded at 490 nanometers using a spectrophotometer in Dr. Robert Letcher's lab. The absorbances were then plugged into this equation here from Garcia-Martin 2019b, which shows the relationship between dissolved oxygen consumption, or CrO2, and the absorbances from INT reduction, INTR. This equation was crafted by comparing the oxygen consumption method by Winkler and the cellular respiration rates by the INT method. This was done with 2 species of dinoflagellates, 1 species of diatom, and 2 species of coccolithophores, including E. huxleyi. They were also found to be comparable methods, and we can use this linear trend and this equation to actually calculate the amount of oxygen consumed by the INT method. Moving on from my lab experiments, I wanted to discuss my collection and analysis of both surface and subsurface water samples on the East Coast Ocean Acidification Research Cruise, which sailed throughout the Gulf of Maine, as represented up here in Ocean Data View, in August of 2022, was the portion that I was on. I was able to measure respiration rates both with the Winkler method and the INT ETS method. While the Winkler samples are still being processed, when I was at sea, we compared a couple of the respiration rates I measured with INT and some of the Winkler samples, and at the time, they were comparable. A unique aspect of measuring respiration with the INT method is the concept of size fractionation. To do this, we want to determine what sizes of phytoplankton and zooplankton were responsible for respiration. So first, we have our 2 filters set up. So the top filter here was equipped with 0.8 micrometer filters, and the bottom filter there was equipped with a 0.4 micrometer filter. The larger size was meant to catch clumps of phytoplankton, zooplankton, eukaryotes, zooplankton fecal pellets, and marine snow, while the smaller size was to catch individual phytoplankton cells. It should be noted that when we are considering which size is more representative of larger clumps of phytoplankton, that's going to be your 0.8 micrometer size. We will be getting back to this. On to my results. I want to first discuss my findings from the Gulf of Maine research cruise. Samples were collected both at the surface, approximately 5 meters in depth, and at the subsurface, about 15 meters. I will say, because of this, we had quite a lot of samples, and it was a very time-consuming process. It took between 4 and 12 hours to filter one sample, so it was a very time-consuming process. So what we're looking at in this first figure here is all of the stations that were filtered at the surface level, or about 5 meters in depth. So here, our color bar indicates the percent of the net community respiration that is dominated by the size class that is representative of clumps of phytoplankton. The darker purple and bluish colors are only a little bit dominated by the larger size fraction, whereas the reds, yellows, and greens are very highly dominated by this size. Additionally, the net community respiration rates, the volumetric net community respiration rates were plotted here, indicated by our black circles. So the larger the circle you see here, the larger the cellular respiration rate. So this station had a very high cellular respiration rate compared to this one, would be an example. So what does this graph actually show us? So here we can see that about 23 out of the 30 stations had over a 50% dominance of the larger size fraction, corresponding to our large groups of phytoplankton. Additionally, I did want to compare these findings with another study, Garcia-Martin et al. 2019A, which was looking at cellular respiration rates in the Atlantic, including areas where we had oligotrophic waters. Now, this means areas with lower nutrients, and there's going to be less productivity and less respiration there as well. So the Gulf of Maine is a very productive region, and we expected the rates to be quite a bit higher for this sample than the Garcia-Martin 2019A paper. And we did find this to be true. Spoilers, both at surface and depth, as we'll see in a moment. But the Gulf of Maine had on average between 60 and 423 micrograms of oxygen per liter per day. While in the oligotrophic regions from the Garcia-Martin et al. 2019A paper, it was closer to 7 to 137 micrograms of oxygen per liter per day. Additionally, I wanted to mention one of the things we were looking at is how does respiration change based on where you are in the ocean. We expected that the coastal areas would have higher rates of net cellular respiration because there's a higher nutrient concentration in those areas. Should be noted that we did have quite a few stations in coastal areas that had higher net community respiration rates, but there were also stations out in the open ocean that were higher rates than we expected. Moving on to the subsurface samples. So this was at approximately 15 meters in depth. Again, your color bar indicates the dominance of the larger size fraction representing phytoplankton, and the black circles indicate net community respiration rates. Again, we can see here that 23 stations were dominated by that larger size fraction that's representative of phytoplankton. As well, we see that the Gulf of Maine did have those higher rates of cellular respiration compared to the oligotrophic areas of the Atlantic. So we had rates that were between 99 and 420 micrograms of oxygen per liter per day, whereas in the other sample, it was 22 to 76 micrograms of oxygen per liter per day. Additionally, one thing that is very interesting is, as you can see here, some of the stations became, they were initially dominated by the smaller size fraction, and now they're dominated by the larger size fraction at depth. So one example of this would be this station here. You can see at the surface, it's much closer to being 50-50, whereas at depth, it's clearly very dominated by that larger phytoplankton size. Additionally, this station up here was again close to that 50-50 and got to much closer to about 60-70% being that larger size fraction. Going back to my lab experiments, one thing we were investigating is how phytoplankton growth is impacted by warming waters. According to Edwards et al. 2016, when phytoplankton have access to enough nutrients and light, they will experience increased growth rate at higher temperatures. Leles and Levine 2023 also found that some phytoplankton species may evolve with warming temperatures to develop larger sizes and faster growth rates. As you can see here for Phytoceras tenuissimus samples, the growth rates actually decreased at warmer temperatures. So the 30 degrees Celsius had a much lower growth rate. This is easier to see the comparison between the 22 and the 30 versus the 19 and the 22, but it was a much lower growth rate at 30 degrees Celsius. This is actually consistent with a different paper from Jin and August 2018 paper that specifically C. tenuissimus is able to actually do a trade-off between cell growth and respiration rates, where they're able to grow less but respire more with the cells that they have. Looking at the Emiliana huxleyi over here, the growth rates were much lower than Phytoceras tenuissimus. This could be due to a variety of factors. This culture is normally cultivated at 17 degrees Celsius, and it was at minimum the 19. However, it does have a wide range of temperature range. Additionally, the growth rates were pretty consistent throughout the temperature treatments for Emiliana huxleyi. Moving on to our phytoplankton respiration rates. This is what we're really excited about here. So both volumetric and cell-specific cellular respiration rates were calculated for C. tenuissimus and Emiliana huxleyi based on 3 replicates at each temperature treatment. For C. tenuissimus, the volumetric cellular respiration rates increased by 2 orders of magnitude at 30 degrees Celsius. So there was quite a high increase in respiration rate for C. tenuissimus on a volumetric scale. There was a slight increase in average cell-specific cellular respiration rates, but it's a little easier to see the trend in that increase in growth rates on a volumetric scale. It should be noted that these volumetric rates, the cell-specific rates were calculated from the volumetric rates by multiplying the net community respiration rates by 31.9. Looking at our Emiliana huxleyi, both the average volumetric and cell-specific respiration rates increased slightly. As you can see, both in the volumetric rates and the cell-specific, again, it's a bit easier to see this trend in the volumetric rates. Additionally, in another Garcia Martin paper, I wanted to compare these results for you guys. There was Garcia Martin et al. 2019b. They had a very busy year. The cultures were grown in F2 medium. They had a diatom, which actually had higher respiration rates than a coccolithophore. So in our example, our diatoms also had higher rates than our coccolithophore. And it's interesting that in 2019, Garcia Martin found similar findings looking at respiration rates in similar conditions. Now that we have reviewed my results, let's see how they reflect back on my hypotheses. Give you a second to refresh yourself with my hypotheses. So first, we're going to be looking at my fieldwork experience. So again, the initial hypothesis was that net community respiration in the Gulf of Maine in the summertime is dominated by the size class that represents phytoplankton. As a reminder, this is our 0.08 micrometer filter size, or 0.8 micrometer filter size. And additionally, on a related note, that the net community respiration in the Gulf of Maine in the summer differs on both the spatial scale and with depth at the surface and subsurface. So what did we actually find? So for net community respiration in the Gulf of Maine in the summer, we did find that it was dominated by that size class that represents groups of phytoplankton. Additionally, in the summer, we found that there was a significant difference between surface and subsurface for the smaller size fraction. Again, that's looking at just individual phytoplankton cells. There was a difference between surface and subsurface cellular respiration rates. This was found by doing a paired two-tailed student's t-test on Microsoft Excel with a p-value less than the alpha value of 0.05. However, it was found that there was not a significant difference between the surface and subsurface for the larger size fraction, looking again at that size that's more representative of large groups of phytoplankton. And this was found using another paired two-tailed student's t-test with a p-value greater than 0.05. Additionally, as we talked about a little bit earlier, there was quite a bit of spatial variability, both at the surface and the subsurface. We had stations in the coastal ocean that had very high respiration rates, but we also had stations in the open ocean with high respiration rates. Going back to my lab experiments here. The initial hypothesis was that increasing water temperatures will lead to increasing rates of phytoplankton growth and respiration. This was tested with our Cattosaurus tenuissimus and our Emiliana huxleyi. So for C. tenuissimus, we found a decrease in growth rate. Again, that is consistent with the paper that referenced the trade-off between growth rate and cellular respiration rate, which we did find to be the case here, that the growth rate decreased, but the cellular respiration rates increased. We also found that Emiliana huxleyi had pretty consistent growth rates, and this may have been due to either external factors when we initially cultivated our cultures, or there being a wide temperature range for this phytoplankton species. Again, that 2 degrees to 27 degrees being optimal ranges. We did find that for C. tenuissimus, the average net community respiration values at 30 degrees Celsius was 2 orders of magnitude higher than other temperatures, which was quite a large increase. For Emiliana huxleyi, there was a slight increase in the average net community respiration rates. I will say one thing that is important to note. Again, our 30 degrees Celsius treatment had more realistic light conditions. If I was to do this experiment again, I would want to make sure that all 3 treatments, the 19 degrees Celsius, the 22 degrees Celsius, and the 30 degrees Celsius, would have the 12 on, 12 off light cycle. For future research, I think this research is super important and it was very useful. In the future, I would like to perform more temperature experiments under the consistent light temperatures 12 on, 12 off, with both cyanobacteria and dinoflagellates, as I have already covered the other 2 major phytoplankton groups. I would love to compare the results of seeing cyanobacteria and dinoflagellate respiration rates and compare them to our diatom and coccolithophore. I also believe that it's really important to add INT incubations with size fractionations to future East Coast ocean acidification research cruises, because then we can see the trends in what sizes of plankton are responsible for the respiration and see how that changes with warming waters. I also think it's very important to look at sampling the stations that we sampled in the Gulf of Maine at different times of year and doing the size fractionation as well. We did sample pretty closely after a diatom bloom in the Gulf of Maine, and it was relatively dominated by diatoms. There was also a lot of dinoflagellates in the water at this time, and I would like to see how this changes seasonally. Respiration is an indicator of carbon budgets in marine systems. 34% of respiration in the open ocean is performed by phytoplankton. These phytoplankton play important roles in the biogeochemical cycles within the ocean. By studying phytoplankton metabolism via net community respiration, we can use these data to predict biogeochemical responses to climate change in the Gulf of Maine and in the global ocean. There are very many people I would like to thank today. I first would like to thank my thesis advisor, Dr. Ziervogel, for your continued support. My committee members, Dr. Elizabeth Harvey and Dr. Joseph Salisbury, you 2 have been wonderful mentors, and I really appreciate the time that you took to make sure I have a thesis I'm proud of. I also want to thank Dr. Robert Letcher. Unfortunately, he couldn't be here today, but he shared all of the lab instruments, pretty much quite a variety of the lab instruments in addition to Professor Harvey, and I really thank him for that. Additionally, I would like to thank the UNH Department of Earth Sciences for funding my graduate studies, all of the Ocean Process Analysis Laboratory, thank you for those of you who could be here today, and my fellow Ziervogel lab teammates, Astrid, Zoe, Amelia, and Misha. I also would like to thank the NOAA Ocean Acidification Program and the scientists and technicians from the East Coast Ocean Acidification Cruise. It was a wonderful way to get started in my master's research, and it has been such a blast. Lastly, there are too many people personally that I need to thank, but I do want to take a moment to thank my wonderful parents who are sitting in the back and my partner Brian, who is on Zoom today, for sticking with me through thick and thin and always listening to me talk about my cute little guys. Thank you all for coming today, both in person and virtually. I really appreciate having you here today. I'll now take time for questions.
Thank you, Lydia. Any questions?
At the risk of being very naive, I will ask a question. Thank you.
Yeah.
So you had a couple of before and a dinoflagellum.
And a diatom.
Diatom,
Mhm.
sorry, yeah, so I'm getting confused already. But
That's right.
how much are these specific species of those types of phytoplankton transport that can be generalized, or do you need to do the same thing for all other species of couple and diatom?
It would be ideal to study all of the individual species possible. However, Amelia huxleyi specifically has been studied a lot, and it actually has been, it was part of the development process for a lot of the equations and things you saw up here today. So EHUX can definitely be used to draw conclusions based on other coccolithophore species. Specifically, C. tenuissimus has also been studied highly, not quite as much as Emiliana huxleyi, but they are very widespread in the global ocean, so it's a good example species to draw conclusions about the global ocean itself. Again, if I was to do this experiment again, I would have a lot more different phytoplankton cultures, but unfortunately in only 2 years, that was a little tricky, but definitely EHUX is a great indicator of other coccolithophores. C. tenuissimus has a really wide distribution, and that can be used for a lot of different trends in the global ocean. Yeah, great question. Yeah.
So projecting this out, like, as if I was running an NPZ model or trying to understand the ocean,
Mhm.
would I expect, is there any way you can interpret from your work whether change in respiration would be dominated by change in rates within species or change in the species assemblage within the ocean?
That's a great question. It definitely is going to vary between like the species composition in the region. I think that is likely going to be the main influencer there. Yeah, that's what I would recommend. I do have a Zoom question.
Oh, I was supposed to.
That's okay, I got it. Ben Lafreniere, what factors determine the station locations? Are there areas you were interested in sampling that you were unable to? That is a great question. So I didn't choose the stations. These are stations that have been sampled in ACOA for the past 8 years, I believe, 10 years. So I don't specifically know why the exact stations were chosen. We did sample approximately every other station on the cruise just for time's sake, because we had the 2 depths and the 2 size fractions, and it took at least 4 hours to filter the samples. But there was a wide variety of coastal and open ocean samples, or stations rather. So I assume that was mostly just trying to get as many stations possible representing both the open ocean and coastal areas. That'd be something Joe could answer a little better than I.
Do you want me to answer that?
Sure.
Okay, so, I don't know if the Zoom person can hear, but, the way that we select the, stations in the Gulf of Maine is different than we select them anywhere else. This is a cruise that goes all the way down from Miami, Florida, all the way up to the Gulf of St. Lawrence, and south of the Gulf of Maine, they're all transects. And that's for obvious reasons to get a cross-shelf distribution. In the Gulf of Maine, there's a lot of heterogeneity, there's basins, we want to sample those, there's George's Bank, we want to get up on those very productive coastal regions and banks, off the Scotian shelf and whatnot. So it looks a little more random, but it's meaningful. There's a lot of thought that's gone into it over the years on where to sample.
Yeah. Cool. Other questions? That was the only Zoom one I see right now. Other in-person questions? Yeah, Amelia.
At what depths did you sample at and why?
So we sampled at approximately the surface, which was about 5 meters in depth, and then the deep chlorophyll maximum, so the area where there's going to be the most chlorophyll. This varied station to station. And if you notice, going back to my figure here, we actually had stations where they were too shallow to do a deep chlorophyll max, so we had less stations at greater depth. And the deep chlorophyll maximum varied between like 5.3 meters and closer to about 50 meters. We ended up, on average, it was approximately 15 meters, so that's what we used for our analysis of that subsurface layer.
Did you do in well mixed stations where there was no deep chlorophyll max, like the one on top of George's?
Yeah, for those stations, we just sampled at the surface. We unfortunately couldn't make a comparison there. Any other questions? Yeah.
This might be sort of related to Jamie's question, but I was wondering if you could fill me in on sort of some of the, that was a very high level question. I'm not an oceanographer, so a more basic question, could you connect the lab results and your field studies? Like, if your lab results, and I know that there's so many caveats on doing any lab study.
Yeah.
And your lab study, of course, there's limited, replicates, limited temperatures, but you did see that there was a temperature, some sort of a temperature effect on the respiration. If that bears, at least with one of your species, if that is borne out, what might you expect to see differently in the field if you were gonna go back in 10 years, 20 years, 2100?
Yeah.
Fingers crossed,
Yeah.
if you're gonna go back in the future and look, what would your lab studies predict that you might see differently?
Yeah, that's an awesome question. So based on my lab studies, seeing generally that increase in respiration rate, at least with those two species, it's predicted that if we were going to go out in 10, 20, 100 years in the Gulf of Maine, we would also see higher respiration rates than we currently saw both at surface and subsurface, especially because the larger size fraction was dominated by those larger clumps of phytoplankton. And those were very influential over the total net community respiration rate in the Gulf of Maine, both at surface and subsurface, that these would likely be higher respiration rates in the future than what we are seeing right now. Additionally, we might see other phytoplankton species moving into this environment because there are better water conditions temperature-wise than we're currently seeing. And so that could change what size fractions we're seeing more of in the future. But primarily, I would say that in the future, we'll be seeing higher rates of respiration overall in the Gulf of Maine. And I'd love to take a look at looking at the Gulf of Maine in other seasons. I think that would be really interesting, seeing how these values vary seasonally, especially with the composition of which phytoplankton are more dominant in the area. Yeah, great question. Yes.
So recognizing the fact that your lab work is static samples as opposed to in the field tidal, what correlation could you draw from the samples that you've developed in the lab and the data that you've gathered? And how would that be impacted by tidal activity in the field? How tightly can you see the correlation between the lab results and what one would expect to find in the field?
Thank you guys for coming. I know you guys have to run. I appreciate you being here. Thank you. Yeah, that's a good question. So again, I would say primarily those respiration rates are going to be increasing. Specifically tidal activity, I mean, if there's been like recent storms or something that could change where the phytoplankton are actually located or how much nutrients are in the water in the presence of phytoplankton, but I would say overall seeing that increased respiration in those areas. Any other questions? All right, let me check and see if there's any on Zoom. Looks like we're all set. Well, thank you all so much for coming in today. I really appreciate it.
Yes, thank you.
Get on Zoom.
All right, so the committee will stay